Okay, so let's get right into it. Imagine a global health crisis just appears out of nowhere. There's this invisible culprit, and it's leaving a devastating trail. But for a really crucial period, we had no way to even identify who was carrying it. Well, that's what we're talking about today, the detective story of HIV testing.
Our story really kicks off here, in the early 1980s. In '83, scientists finally put a name to the face. They identified the HIV virus as the cause of AIDS, a huge, monumental breakthrough. But, and this is the key part, look at that next date. It wasn't until 1985 that the first test was actually available.
I mean, really think about that for a second. For 2 whole years, in the middle of all this growing fear and uncertainty, doctors, patients, blood banks, nobody had a reliable way to see the virus. It was a period of just immense worry, and the need for some kind of diagnostic tool was absolutely critical.
And that, right there, brings us to the central question of this whole investigation. How on earth do you develop a test to find a virus that's literally hiding inside the human body? Well, it turns out scientists developed 2 brilliant and very different strategies.
Think about it like this. If you're trying to find an animal out in the wild, you can either look for its footprints, the signs it leaves behind, or you can try to find the animal itself. Well, HIV testing works in a really similar way. We can either look for the body's reaction to the virus, its footprints, or we can look for pieces of the actual virus. Let's start with those footprints.
So this first method is by far the most common approach, and it's all about detecting that trail the virus leaves behind in our immune system.
So what exactly are these footprints? Well, they're called antibodies. When HIV gets into your body, your immune system basically goes on high alert, and it creates these custom-made proteins designed specifically to fight that invader. Now, they don't actually destroy the virus, but their presence is a crystal-clear signal, a fingerprint, if you will, that HIV has been there.
And this slide just shows you how incredibly accessible that detection has become. The rapid antibody test is just a marvel of simplicity. You need a tiny sample, a little bit of a chemical to get the reaction going, and then you just wait a little bit. The test isn't looking for the virus, remember, just for those telltale antibodies.
Now, there are a couple of main types of these antibody tests. You've got the rapid tests, which we just saw, that gives you results in minutes, pretty much anywhere. Then you have the ELISA test, which is kind of the lab-based workhorse. It takes longer, but this is also where we get into 2 really important ideas: sensitivity and specificity. Think of sensitivity as a test's ability to find what it's looking for, to correctly say yes when the virus is there. Specificity is its ability to ignore what it's not looking for, to correctly say no when it's not. Lab tests like ELISA are the gold standard because they're generally super high on both.
But there is a catch when you're looking for footprints. It takes time for your body to actually produce enough antibodies for a test to see them. This delay is called the window period. And here's the crucial point. A test taken too early during this window could give a false negative result. The virus is there, but the footprints just aren't visible yet.
But here's the fantastic news. The earliest tests had a window period of over a month and a half. Thanks to incredible scientific progress, modern tests have slashed that time, making testing way more accurate, way sooner after a potential exposure. Okay, so let's move on to the other method.
Right, so what if we don't want to wait for footprints? What if we want to catch the suspect red-handed? Well, that's where the second category of tests comes into play.
This approach is totally different. It completely bypasses the immune response and just directly searches for actual components of the HIV virus. It's not looking for clues, it's looking for the culprit itself.
So what specific evidence are these tests looking for? They can target 3 key pieces. There's the P24 antigen, which is a protein from the core of the virus. Think of it like finding a button that's fallen off the suspect's coat. It's very early evidence. Then you've got the virus's genetic material. Proviral DNA is like the virus's master blueprint, which it sneaks into our own cells. And viral RNA is like the working photocopies of that blueprint, floating around in the blood as the virus makes more of itself.
And these genetic tests, usually called PCR tests, can answer 2 very different questions. A qualitative test is like a security camera. It just tells you if the virus is present in the building, a simple yes or no. That's for diagnosis. The quantitative test, on the other hand, is like doing a head count. It measures how much virus is there, what we call the viral load. And that is absolutely crucial for monitoring how well a treatment is working.
So why do we need this sophisticated technology? Well, its most important job is for a very specific group of people, for whom those footprint tests, the antibody tests, simply do not work.
And here's why.
During pregnancy, a mother passes her antibodies onto her baby to protect them after birth. It's an amazing part of nature. But it means a baby born to a mother with HIV will have her HIV antibodies, whether the baby has the virus or not. So an antibody test would come back positive, but it could be a false positive. Virus tests solve this whole puzzle by looking directly for the virus, giving a clear, life-saving answer. And this is often done with a simple dried blood spot test. Just a tiny heel prick from the baby is all that's needed.
So there we have it. Two powerful strategies, looking for the footprints and looking for the virus itself. And together, they form this comprehensive toolkit for detection, diagnosis, and care.
Let's just do a quick side-by-side to lock all this in. Antibody tests, the footprint hunters. They find the body's immune response. They have a longer window period, and they're perfect for routine screening. Virus tests, the ones that find the animal. They detect pieces of the actual virus, have a much shorter window period, and are absolutely essential for early diagnosis, for testing newborns, and for monitoring treatment.
And that toolkit has completely transformed public health. I mean, what started as just a way to diagnose sick people and protect the blood supply has evolved into so much more. Today, testing is a cornerstone of prevention. It helps people keep track of their treatment, and it gives vital data to researchers. It's an active tool for managing health, not just a passive diagnostic anymore.
You know, we've come such a long way from flying blind. The science has given us this incredible array of tools to find and manage HIV. The detective work of the scientists is, for the most part, done. So the question that remains for all of us is this: how do we make sure these tools are used effectively, equitably, and compassionately for absolutely everyone?